Hello, everyone. It's a great pleasure to introduce you a new direction in my lab, so in which we use the nanopore sequencing to sequence the to detect the structural variations that in diverse domestic chickens. So a lot of you may have heard about the SNP, single nuclear polymorphism, so which actually are changes, mutation that only affect one base pair. So for the mutation that affect over 50 base pair, we call that the structural variations, or short for SV. So human actually have around each human have around the 20,000 structural variations because they impact more base pairs. So they, if you count the region they affected, so they actually affect more than the single nuclear polymorphism. That's why so a lot of the phenotypic variations and uh, the disease that inheritable diseases that cannot be explained by SNP. So it's believed to be explained can be explained by the structural variations, and so they call that misinheritability. So these kind of structural variations, so there's a different format, like you have deletions, insertions, tandem duplications, intersperse duplications, inversions, and translocations. So they actually affect the karyotype. So these kind of variations exist in the populations within a species, but through time, so they will be fixed, and so then evolve to be a separate species. So if you look at the current domestic animals, you can see like their chromosome numbers varies dramatically, like their karyotype. So for example, like the cat has 38 chromosomes, the camel has 74. So this is in mammals. However, if you look at in birds, in avians, their karyotype is unusually stable. So majority of the bird species have around 80 chromosomes. If you look at their karyotype, they're almost identical, have some large chromosome and have a bunch of um, microchromosomes. So this unusual stable karyotype is believed to be due to a low activity of their transposable elements. Transposable elements are jumping elements, so which is a major force to create the structural variations. And as you can see that compared to mammals, chicken has very low amount of the transposable element. However, like even chicken has very low TEs, but domestic chicken exhibit extensive phenotypic variations. We know like current model chicken are domesticated from the red jungle fall about 8,000 years ago. And currently the two main applications like for the domestic chicken are one for egg layer, two main type. One is egg layers, one is meat type chickens. Of course, there's a lot. This is industrialized chicken, but there's also many thousands of village local lines. So they all have very diverse phenotypic variations. So then the question is that given their very stable karyotype, but they have very diverse like phenotypic variations, then do chicken have extensive structural variations? And so it is believed that chicken has very low TE activities. What exactly is the remaining activity of TEs? And where are these TE insertion localized? Do they contribute to any of the phenotypic variations and any functions? So we choose six representative chicken lines from all over the world, that including like a layer white Leghorn meat type chickens that was set up like in Georgia, and then three local lines, one from the South America, Araucana, and 2 farm chicken. One is a black bone chicken, one is Tibetan chicken that is adapted to the high altitude. And also we sequence a line of the red jungle 4 that is maintained in Davis that has been used for chicken genome sequencing. So one reason, like we know least about the structure among all the mutation type, we know least about the structural variations. It is because that genome often have repetitive regions, and so the conventional sequencing is a short read sequencing. So the short reads actually cannot be uniquely mapped so at the repetitive region to the repetitive regions. That's why so the genome often have these form as a germ assembly only often form like a contigs because the gap region are often repeat regions which cannot be uniquely mapped. So one solution that is the new methodology called the Oxford Nanopore sequencing, in which like a single molecule nucleic acid strand got pumped through a pore, and then as the nucleic acid went through the pore, so the current would change, and then you can interpret the sequence from the current changes. So that actually do not have any limitation on the sequence length. So this kind of long reads can span the repeat. So that's why can provide a pretty comprehensive and accurate mapping for the structural variations. So we use these long Oxford Nanopore reads to sequence the 6 chickens from each breed. And we can get the mean length of the read is over 10 kb. And the maximum length you can see is almost reach 500,000. So when we do the mapping, we find that like over 80% that could be mapped to the genome. And the error rate is around over 10, but the depth is pretty high. So that can be used to eliminate the high error rate. We have over 30-fold coverage. So overall, we have got very high quality of Oxford Nanopore reads in chicken. So this allowed us to define structural variation in high confidence. We use very stringent cutoff when we define the variations. As you can see that each chicken so have around 1,700 structural variation when compared to the wild chicken reference genome. And not surprisingly, because the reference genome is from wild chicken, the red jungle 4 has only have around 5,000 structural variation. So it's lower than other domestic chicken, but that also indicates that the population still have the genetic diversity there within that population. And the majority of the structural variation come from either insertion or like deletions, and have some duplication that inversion have the least abundant. So then we want to know how much these variations contribute by the transposon. So then we start to look, start from these over 10,000 insertions, and we blast that to the TE consensus sequence. So in chicken, so there's actually very diverse TE families in chicken genome that it can be mainly categorized to 2 groups. One is DNA transposon, one is retrotransposon. So retrotransposon, their replication require transcription. And the retrotransposon can be further divided by the long-term repeat LTR transposon and non-LTR transposon. The non-LTR transposon, which is can be further divided by the to line element and sign element. In chicken, sign element are mostly missing, and the line and DNA transposon is believed to be pretty ancient. And the most active one, the recent one, is believed to be long LTR retrotransposon. And they all come from that retrovirus invasion. So they are endogenous retroviruses. So then we find that we use multiple filter, and in the end, we find around 500 transposition integration sites. These actually include the known insertion that lead to the blue shell eggs in Araucana. So, and all these integration transposition are coming from only 30 active 30 T families. So they are include 29 endogenous retrovirus and 1 line element, CR1. So then we want to know where are these transposon localized. We find that around 44% of them falling into the genic region, with that half is in the mRNA region, half in the link RNA region. And then we find the majority of the insertion for the mRNA and link RNA are in the intron region, with only a few, like a small percent, like in the axonic region. And we also find that there's genes, some genes, 6 genes are independently inserted twice. So that's suggesting that could be some functional relevance for these insertions. So we look at all the genes that harbor the transposon insertion. So we saw that it would be growth would be the gene that would be mostly affected, as that is the one trait that has been artificially selected. But surprisingly, we find that like almost the most gene got affected are the gene related to neurological functions. A lot of them are known to, in the human, known to associate with autism. So consistent with our finding, we find that the genes that are inserted by TE are expressed in the brain the most. We can detect them highly expressed in the brain and cerebellums. So then we can conclude here is that the domestic chicken have pretty extensive structural variations around a similar level as in human. And we can detect, although the only small percent is affected by TE, so we find 29 retrovirus and 1 line element that's still active in chickens. And we find the TE insertions mostly disrupt the gene with neurological functions. So I would like to conclude here and thank the people who did the work, including you in my lab, and also it's a collaboration, very fruitful collaboration with Xin Li in McGill University, and also the analysis we have great help from the Chi from the Ohio State University, and this is supported by the USDA grant and with some stretch of money from the NIH grant to support this expensive sequencing. I would like to end here and take questions.
Thank you, Dr. Li. Very interesting. And we do have a question. How much would the addition of these structural variations add to poultry breeding programs in terms of genetic gain in production parameters?
Can you hear me well?
Yes.
Okay. So that's what we have in the system. So we have only analyzed the insertion, the T-induced insertions, because overall there are over around 17,000. So we haven't. So for the duplication, deletion, inversion, we haven't looked into detail about their functional impact yet. We assume we expect there will be a lot, but we need to analyze the association. Probably we need to include more different independent developed domestic chicken to find the association of those structural variation with the traits.
Thank you for that. I was thinking about that myself coming from the commercial sector, and I just thought, particularly thinking about broiler chickens, how would that work?